while the session is going on, do well to join us. In our subsequent section, we're going to be having podcast sections, and the Telegram app we'll be going to use. And we share most of our information and documents and resources on the Telegram platform than on the WhatsApp platform. So we would like you to join the Telegram platform, which is our official platform. It equally doubles as our library, our virtual library. So all information and resources you need on the General Club can be accessed on the Telegram platform. So without much ado, I would like to introduce quickly the speaker for today. Is going to help us to review this article on QSAR-based virtual screening of traditional Chinese medicine for the identification of mitotic kinesin E5, EG5 inhibitors. This is more or less for those of you who are in herbal medicine. And I think it's something that is also quite relevant to all of us. We are in Africa, and we know that most of the herbs that you and I tend to overlook contains a lot of medicinal properties and polyphenols that are used by these Western biofarmers and also big farmers to manufacture different medications and then export it back to us to buy. So he's going to give an insight as to how in some of these medications, some of these herbs can actually serve as a reservoir of these nutraceuticals and these minerals that could be useful in pharmaceutical production. Without much ado, I'd like to introduce the speaker. The speaker is Dr. Bodan Damilola, who is undergraduate, so he's an undergrad actually at the Adinkole Adjacent University, who majors in biochemistry. He currently has 6 publications in reputable journals. His research areas focus on molecular and computational biology. And so before we start, I would like to call on our coordinator, Dr. Duhati, to briefly welcome us, and then we can kickstart the section. So Dr. Duhati, if you are here, the floor is yours. Dr. Fumilayo Duhati, are you here? Hello, Doc. Okay, I'm sure Doc may be offline at the moment. So she will do that at the tail end of our section. I'd like to acknowledge all my colleagues and who've been able to put this together and make this to reality. Dr. Anio, thank you very much for always anchoring this and taking the lead on this.
Thank you, sir.
Ravi Chandra, Ravi, thank you for joining, and thank you for the support you continue to give to the leadership.
Thank you. Thank you, Dr.
Thanks everyone. Thank Dr. Felix, who behind the scene does most of the things on our statistics and data analysis. So without much ado, I'll call on Bodan Damilola to lead the section. Damilola, the floor is yours.
Hello, everyone. Good evening. I hope we're all doing well. My name is another graduate at Adinkole, like just saying. And today I'll be taking you all in a review session based on a recent publication. So first and first is I'll be introducing you guys. And before I start, I want to thank Dr. Anio and Dr. Fumlayo for the opportunity. It's really, really great. This has been really, really helpful to me. Thank you very much. And thank you to everyone, to the whole participants. So thank you. So please, hope you all can hear me.
Very well, Bodo.
Okay.
Yes, we can hear you. You can carry on.
Okay. So so the title of the journal is QSAR-based virtual screening of traditional Chinese medicine for the identification of mitotic kinesin EG5 inhibitors. So basically, I'm just going to break down these keywords to explain everything that was done in this journal, in this publication. So the first thing is that everything we did here in this publication, we try to use an in silico method. So in silico in term means computational approach. So instead of going to the lab and try to look for an inhibitor, or going try in vitro and in vivo methods, we try what use what's computational approach, which is better and it saves time and is even saves money as well. So that's the major goal of this thing, to identify what potential human EG5 inhibitors. So so the first thing first is what is mitosine kinesin EG5? What is that protein? Or what is that enzyme? So this enzyme is really important. It plays a very crucial role in the process of cell division by separating chromosomes and moving them to the opposite poles of the cell. This, which is a really, really important step in what in cell division. And we know cancer cells, since they will often make what's rapid and rapid cell division, we have to what prevent that step. We have to prevent that step to prevent cancer cells from undergoing that important step in what in progressing or in proliferating. So there's an enzyme involved in that. The enzyme involved in that step is what mitotic what kinesin what EG5. So or human EG5, that's another popular name. So the thing here is we have to inhibit this enzyme. This enzyme is causing us trouble because cancer cells, once they see this enzyme, they proliferate and they become they expand and become abnormal. So that is what we wanted to use. That's the target we wanted to use. And the way we wanted to find a cure from is traditional Chinese medicine. And recently, or in the past, artemisinin, which is a very, very, very popular anti-malaria drug, was found from what? This group of compounds called traditional Chinese medicine. There have been a lot of studies, quite a lot of studies that have been trying to trace and trying to find out the therapeutic effects of different traditional Chinese medicine compounds. So I applied that in this study. I got over 3,000 traditional Chinese compounds, or 2,000, over 2,000 traditional Chinese compounds, and I try to find which one is the perfect inhibitor to what to G5. So there's a Chinese I said, as I said earlier, artemisinin was discovered by Professor Tu Yoyo, which won a prize in physiology 2015. So, CUSA based screening. So, once you are in the computational world, I think there's a term called virtual screening that you should be getting adapted. You get adapted to, and virtual screening really just means
Sorry,
we have.
sorry,
Hello,
sorry,
sir.
sorry.
Hello, sir.
Please, if okay, alternatively, if that doesn't happen, I'll go through the chat. I'll go through the contact details and I'll mute you without your permission. Please, we need to have an orderly section. Okay.
Oh.
Mike, so have an orderly section. All right. Please carry on. Thank you.
Okay, sir. So I said CUSA-based virtual screening. This is a very, very popular computational term everyone will come across if you are into that field. And the term just mainly just based that if we have a standard, or if we have a parameter, then you try to screen a library of compounds based on what that parameter. So any compound that doesn't fit into that your parameter, you filter them out. So that's exactly what CUSA based virtual screening. So that is virtual screening. So now CUSA based. So the virtual screening that we did, or the filtering we did, was based on what? Based on what? The CUSA. So, and I think I said something about the CUSA there. So it said CUSA. The meaning of CUSA is just basically quantitative structure activity relationship. So it's just a basic way, or it's a computational approach where we try to relate the biological, or we try to predict the biological activity of a compound based on what? Based on the structural conformation, the structural arrangement. So that is everything of CUSA. So that is the term parameter CUSA. So we checked compounds that that were predicted to have high biological effects based on their structural components, to what? That's what we used to what? To filter out this list of compounds, which were, I said earlier, were over what? 2000. So um so at the end of this study, or when we started this study, we just had three goals, and the first one was we should discover a better inhibitor of EG5. Remember, I said EG5 was a protein, the target, or the enzyme and we should discover a better inhibitor of EG5 and compare it to what? To the standard. So there's currently a standard drug called monastrol. So we wanted to check if our discovered drug is better than what? Monastrol. So then we tried to check, we tried to confirm the binding results or the docking scores through another docking methodology. So basically in this computational approach or in this computational biology field, we have various kind of docking methodology. So okay. Okay. So there are various types of docking methodology. I think the popular one is the rigid receptor docking, where we try to take the ligand binding to the active site of a protein as a rigid structure. So that's like another story for another day. So we tried checking another methodology, which is the QPLD. That's a quantum-based and quantum polarized ligand docking, where we try to make we treated the ligand as quantum and the body as mechanism, something like that. So that's what we did. We tried to confirm the first docking score based on the rigid receptor docking, then using what? Quantum polarized ligand docking. So, and the third thing was we should reconfirm these results again. So we got the results in number one, in number two again. Then we should reconfirm these results again using what we call molecular simulation or MD simulation. This is where we try to predict the stability of a particular drug in the active site of a protein. So that was that was our aim in this study, and I think we did that. So, well, how did we do it? I think that should be the question now. So the first thing first is every drug you take, every pharmaceutical drug or every pharmaceutical drug we all use in our daily life, they are orally bioavailable. That means they can be they are that means they are everywhere, or how let's say they are orally bioavailable in the body. Every drug you take, there are 100, there's a percentage we rank it say, so 100%. But the basic thing is that for you to have a bioavailable drug, a bioavailable compound, or for a pharmaceutical, for a drug to come out from the pharmacy, they have to obey a rule, what we call the basic rule of five. So that's what I wrote ROV. That is rule of five. So rule of five is equals to 0. So rule of five is just a basic rule where a set of four rules where we believe every drug should obey. Every drug should obey. So from this study now, we see N is equals to 256. That means I started with 256 compounds. So I screened out any compound that did not have up to that disobeyed any of the rule of five. So any compound that disobey or that doesn't obey any of the rule of five, that disobeys any of the rule of five, I throw it out. So I filtered them out. So after that, 256 were reduced down to 1,637. So now, after reducing that, I predicted the biological activity of these compounds using a QSAM model. So anyone with a biological activity value, which is called PIC50, less than 5.768 nanometer, um, I think that's it, nanometer. So that is it. So anyone less than that, I filtered them out again. So which is gone. So from 167, we're left with okay. So after that, I used virtual screening again. Under virtual screening, remember I said virtual screening is you screen based on it. Hello?
Even down to actually, no be small, you know. But
My tape just spoiled.
Hello.
Hello?
Hello.
Hey.
Please, when you join the meeting, mute your mic. You are distracting us. I think next time I will take such people
You go
off.
feel them.
Okay. So remember I said earlier, why I said virtual screening is screening based on the particular parameter. So the first parameter we used here was the QSAM model, right?
It's okay now.
Okay.
Please, can you just mute everybody until Q&A session?
Exactly. I think he kept on owning his own mic. Mr. Muhammad.
Okay.
Please,
So
brother, you can
Yes, so please, you can go ahead. I'll be muting everybody. It's distracting. Hello, Damilola. I think I muted you, so unmute yourself.
Okay, yes, sir. So I said earlier, virtual screening is screening a list of compounds or a group of compounds based on what particular parameters or particular parameter. So now with the first virtual screening, we can see it based on what the QSAM model, where we try to filter anything that doesn't have a biological property predicted to be less than 5.79. We filter them out. Then the next parameter we used was pharmacophore hypothesis. So pharmacophore hypothesis is basically we're generating a hypothesis or a model where we try to predict um, the the features present in a co-crystallized ligand, which is another term. A co-crystallized ligand binding to what? Binding to a protein. So we try to map out the features present in a compound or present in a co-crystallized drug and try to we try to look for these features and know these features that these are the features that made this compound bind to this protein or bind to this enzyme. So after mapping out those features, we use it to filter this compound to again. So the next thing is structure-based structure-based screening, which is also called docking, which is very popular, is a very popular thing. So HCVS docking, which is like a virtual screen, is very, very fast, not precise. So anything with a docking score less than -6.0, we filter them out. So by the way, the docking scores, you will notice it is a -6. So the lower the docking score, the better the binding affinity. So once you see -6, you should understand that it is going -6, -7. So in this term, -7 is way better than -1. So that means it has a very better binding affinity. So after the HCVS docking, we went next to SP docking, which is standard precision. So we repeated that docking procedure again, but this time we're using a different precision method. So we filtered those compounds with a docking score less than -7. So which left us with N 64 compounds. So now the top 8 compounds, we took them to the last precision of docking, which is extra precision. So extra precision is a very, very accurate method, but takes longer time. So we use extra precision to dock those compounds, those 8 compounds. And we had, so from that result, from those 8 compounds we docked, we use what we call MMGBSA. So MMGBSA is like a post-docking analysis where we try to confirm the docking scores, or we try to confirm the binding affinity between the compound and the ligand. On normal days or in certain times, you can find a a compound with a very high docking score, but with a low MMGBSA. So that means the binding affinity doesn't correlate. So MMGBSA is just used to what? To correspond or to confirm that docking score you earlier got. So MMGBSA, we did the MMGBSA prediction for the top 5 compounds. Then DFT analysis. DFT is a computational chemistry skill where you try to predict the electronic structure or the electronic property or the something they call HOMO, lowest unoccupied molecular orbital and highest occupied molecular orbital. So you try to predict the electronic structure. Try to find out which part is the donor and which part is what the acceptor in a chemical structure in a chemical compound. So we did that. We did that analysis for the top 5 compounds again. Then ADMET analysis. So ADMET just really means absorption, distribution, metabolism, and excretion. So we try to predict the ADMET property of the top 5 compounds again. So remember earlier I said quantum, and the objective of this study, the second one was to confirm the results we've got using a different docking approach. So the second docking approach we use is what QMM, quantum mechanism, quantum mechanism docking, which is also called QPLD. So this one is way precise than the other method of docking that I mentioned earlier. So this is precise. So we did this method because it's a very, very computer intensive method. So we just did for the top compound, which is the it compound, the best compound, and we did it for the standard drug, which we used to compare, which is monastrol. Then MD simulation. MD simulation, we try to predict the stability of a compound in the active site of a protein. So we did that for the first compound, the top compound, the second compound, and the standard drug. So that is why N is equals to 3 there. So okay, the results. So the first results that we should say now is this is the figure of the best. So from the traditional Chinese medicine compounds, remember we started with over 2,500 compounds. So these compound 1, 2, 3, 4, 5, these are the best compounds. These are the best compounds. So the first compound, compound 1, compound 2, compound 3, and compound 4. So this is a monastrol. Monastrol is just there. This is the standard drug. Okay, let me see. Okay, so this is the QSAR model. This is like a data from the QSAR model. We trained the data and then we tested it. We tested the data against a particular against our compounds, the traditional Chinese medicine compounds. Okay, so now this is a very, very popular thing we see in computational biology, which shows the interaction of the compound in the active site of an enzyme. So we should remember basically amino acids make up proteins, right? And enzymes are proteins. So these active sites, what is just showing is that our compound 1, this is our compound 1. It is making an H bond interaction with glutamine 116, and it's also making an H bond reaction with glutamine 118. So, and these other green amino acids you can see, they're called hydrophobic amino acids. So, okay, so thanks to the how class organized by Autoid, this was what made me learn this. I had to learn. I created this with Aro. A doctor came online to teach us 3 days in health science. So I really took an interest and I learned quite a lot. So this heat map was made with R. So this heat map just contains the admit property of the top 5 compounds and monasterol, which is standard. So from this result, what you can see is I think the basic thing here should be human overviability. No. Human intestinal absorption. No. So these are just properties that just that we want to know. Blood-brain barrier. That means none of when it is red, that means no. That means none of these compounds can cause the blood-brain barrier. That means they can't be used as any treatment, any drug. It can be used as a drug for any disease related to the central nervous system. So, okay. So I said something earlier about DFT, where I tried to say you try to predict the electronic, so the donor and the acceptor of the compound. So this is something like that. So the first one is the optimized state. This is a compound in an optimized state. And the second one is the compound in its almost state. And the last one is your compound in its low state. So molecular simulation. So remember I said earlier, molecular simulation is used to predict the stability of a compound in the active site of the protein. So now from this result, for you to understand this well, you should know that the blue and the red line should stay together throughout the whole simulation period. So we did this simulation for 100 nanoseconds. Try to predict the stability of this compound in 100 nanoseconds. So the first result you can see there, that's our compound 1. And if you can compare it to compound, so if you can compare it to C, which is our standard drug, you can see it is way better because of the overlapping of the blue and the red line. So once they overlap a lot, that means the compound is what? It is stable in the protein, in the active site of the protein. So we can see a different thing here in the B, where we see the compound 2, which is not stable enough. We can see the gap between the blue and the red line. We can see the gap between the blue and the blue line. Then around these 16 nanoseconds to 18 nanoseconds, it gets back into the binding site, but after it leaves the binding site. So we can see that through our virtual screening process and everything, we've been able to identify a very, very good compound that has a better binding affinity, is better stability than monasterol, which is C in the active site of EG5. So, okay, discussion and conclusion. So I think the conclusion, so everything in this study, we try to use various in silico techniques or different computational method to try to identify inhibitor of EG5. So the results of the molecular dynamic simulation really supported our studies and have further our previous results showed that compound 1, which doesn't have a name, I think it has a Chinese name. I was not able to interpret it. So it does, so I named it compound 1, showing that compound 1 had the better and stable binding affinity to the binding site of EG5 than drug, which is the standard drug. So that is all for me. That is all for me. So, thank you guys for listening.
Thank you very much, Dami.
Yeah, thank you very much, sir.
So stay behind for questions.
Okay, sir.
Anybody with the first question to ask? I don't know, I have a couple of them because yes, I have a background in
Conversation.
Yeah, computational biology and stuff like that. So yes. Okay, my first question is with respect to the docking method you used.
Okay, sir.
So let me put it simply for those of you who may not understand docking. So docking is simply molecular modeling technique that you use to predict how protein, specifically enzymes, interact with small molecules, which we call the ligands.
Yes, sir.
I wanted to find out, yes, you really emphasize on the binding affinity.
Yes,
where
sir.
you need to identify any novel or potent ligands.
Yes, yes, sir. Yes, sir. So I said so the compound 1, which doesn't have a name, it has a Chinese name, but we're not able to translate it. So this is a novel compound that we found that is way better than monasterol, the standard, sir.
Okay. The Chinese name, was it written in Chinese characters?
Yes, sir. This Chinese string was written in Chinese character. Then
So
I'm trying to.
If you can send a character to me, I can get you the name in English. Essentially, so the Chinese, when it comes to the scientific literature, most of them are derivatives from English or other languages. So we can easily get the original names. Yeah.
Okay, sir. Okay, sir. Okay, sir.
Okay. And the heat map, I think you have a very good heat map, but unfortunately, you couldn't really explain it well.
I'll do that, sir. I'll try to send the Chinese name to you, sir, immediately.
Is this paper published?
Yes, sir.
Which journal?
Computational biology and chemistry.
Oh, okay, okay.
Yes,
Okay,
sir.
I think that is my initial question. I have other questions. I'll reserve them and open it for my other colleagues who have questions to ask. So
Okay,
please,
sir.
you can unmute your mic or you send your question in the chat box and I'll read it out for them to respond to.
I think Dr. Felix is here to critique the statistics.
Okay, so Dr. Felix, I understand you are taking the statistics part. So the floor is yours. Hello, Dr. Yam.
Let's proceed if there is anyone that has questions.
Okay. Okay. Any questions? So, okay, let me ask my other questions. Which database?
Okay,
which database did you use? Did you extract your data from?
Okay, for the compounds, they were given to me from my professor. So I could not really get the database source because is for you to have Chinese compounds, you must like for you to have that amount of Chinese compounds, you must like have a particular database. But I was not able to get it from my professor. But for other properties that was predicted, like this admit property, I used.
Hello. Hello. Hello.
Hello, Bodum, I can't hear you.
I'm sure his network tripped.
There's a question in the chat box.
Yes,
So-
I think he's
Okay, I got disconnected. I'll connect
Okay, that's
immediately. Yes, sir. Sorry.
okay. So there's a question for you. Someone is asking which molecular docking techniques software did you use?
Okay, I use the popular and the best one, Mistral software, Scodinja. I think that's a popular name around. Scodinja, Mistral Scodinja.
Okay, can you put it in the chat box, especially for someone who want to try
Okay,
that?
sir. I'll do that now, sir.
Right. Yes, so with the work you've done so far, how do you think it could be applied on our medicinal plants, even though we don't have much molecular data on our medicinal plants that we've screened so far? How relevant would this study be? Do you think it can be to the future of herbal medicine on the continent?
There's something I really talked about, and there's something I was really advised, and that should be even though the compound has shown like great potential via in silico techniques. So I think the next step should be trying it through in vivo and in vitro techniques to confirm that property. But if it fails doing those trials, then derivation should like they should create derivatives of like the drug of the compound to try to improve it in like they should add one or two molecules to remove a particular molecule, add particular functional groups, try to edit the compound to make sure it fits into this thing. So the probability of getting a particular medicinal plant or a medicinal phytochemical to be the actual drug is kind of rare. So, but I feel with modifying and changing its form, I think we are good to go. Like it's a very, very solid thing. I hope so.
Okay, you indicated that the compound is usually found in the Chinese herb.
Yes, sir.
Yes. Is there any herb in Africa that you can trace these compound to? That could be a potential reservoir of this compound? Maybe based on
No,
your literature.
I think no, because the first thing is since we don't have a particular name for the compound, like others, we have camophor, quercetin, isoquercetin, but since we don't have like the original English name, so I think tracing it will be hard. Well, once I get the Chinese name and try to like turn it to, or if I can get, well, just try to get the IUPAC name. So I think it should be possible to try to trace it to other
If
traditional.
we can also get the picture, that is why we have the botanist. And I believe the botanist too can help in identification. Firstly, to get local name by the structure. Once you get a picture of that plant, you can post it for identification, then we'll be able to trace it. If it is available in this African region, then we can pick it up from there.
Yes, ma'am. Okay, ma'am. Okay, ma'am. The structure is right there, but the plants is just the thing because I feel the database just filters out, just brings out the compound. But I will try to look deeper into the data I have here and try to see if I can trace it out or if I can see something relating to a plant, a particular plant, sir.
Okay, I will need a plant. I can help you identify it, especially with the Chinese character.
Okay, sir.
Or even with the photo, just like Dr. Hani, you suggested, we can help you identify that.
Okay, sir.
That is very key. Dr. Ravi. Yes.
Hello.
Yes.
Hello, sir.
Yeah, hello, Mr. Jamilo. Nice presentation.
Thank you very much, sir.
It's in fact a very interesting study on, I think you're mostly focused on the herbal compounds, right?
Hello, sir. Can you come again, sir?
Am I audible to you, Mr. Damilolo?
Yes, sir.
Am I audible? Sir? Yeah, am I audible to you?
Okay, yeah. You're audible.
Yeah. So what I meant is basically your research is mostly focused on identification of herbal compounds, right? So especially active compounds, maybe. Mr. Damilalo, are you able to follow me?
Sorry, can you come again? The network is breaking off. Okay.
Hello, Ravi. If you are using an earpiece, please don't use the earpiece because it gives a feedback. Just speak direct into the gadget so that we can hear you. Because your voice is not
actually, I'm not using the earpiece. Yeah. So
okay.
I'm speaking
direct.
We can hear you well now.
I think it's better.
Yeah, it's better, right? Yeah. So what I meant is like basically you're working on identifying the active herbal compounds, right?
Yes, sir. They are identifying their active compounds.
Yeah.
Yes, sir.
So you're trying to segregate the chemical constituents, mostly active, compounds. So what I meant is basically you can consider it like probably a suggestion. What I meant. So are there any do you have any idea, like for example, where was your study site? So I'm really curious to know, like where did you did the collection and all of the herbs? Is it back in China?
No. As I said earlier, the collection of the herbs, it was just a list. It was just an SDF format of compounds, and it was titled Chinese medicine, traditional Chinese medicine compounds. So I got it from my professor at Ado Ekiti. So he was working on it. So I accessed.
Okay, so do you have any idea, like where are these specimens collected, or where are this? So any idea, like where these have been?
I really don't have any idea.
I think that would be interesting find, because if you find the same species, similar species in Africa,
yes.
So I mean, at least like genetic composition,
Okay.
So probably you can segregate the same kind of chemical compound So which has the similar effect. I think that would make a really good study, good comparative study.
Yeah, exactly, sir. Thank you very much, sir.
I think
I'll try to.
So just my kind of suggestion, in this. So I thought because probably most of the Chinese traditional medicine, they usually collect herbs from different East Asian and South Asian countries. So I guess.
Oh.
So okay. Where India also comes like mostly from Himalayas and all. There are lots of herbal medicine, medicinal plants. So what I meant is like basically if you can really look at the difference between or maybe the similar compounds, if you could locate in Africa, especially with similar geography and ecology, I think that would be really good study. Because since I think your research has a good potential, probably where you can take it forward.
All right, Ravi. So I think yes,
Okay.
suggestion is well taken.
Thank you. Yeah.
Yeah.
Okay. Okay.
Um,
Okay.
there's another question here. I think you already responded to this partly. Did you use Chinese database or PubChem? The PubChem database. I think you indicated it's a Chinese database, but you don't know the name. Hello? I think he dropped off again.
Yes, sir. Sorry, the network. Network.
Yes. So someone is asking trying to confirm the database you use. Was it a Chinese database or a Russian database?
Exactly. So I've been saying this.
Yeah.
The normally when I try to work on any plant, the first database I work on is PubChem. PubChem is like my go-to database that I use.
Yeah.
But for this case, I did not use PubChem because I was just handed over the file, which contained the SDF format, which just contained all the traditional Chinese compounds. So the source, the real source, I was not able to get because I could not really ask my professor. That's the thing, sir.
Can I
But
ask who shared the files with you?
okay, so my professor at the lab, sir.
Is he a Chinese?
No, he's a Nigerian. Well, I think
Okay.
it works on traditional Chinese compounds, the same thing, phytomedicine.
Okay. I'm sure you can pose these questions because it's really relevant to get to the source, to know the source of data.
Yes, sir.
So that when you are speaking, there's any issue, you can trace it to the source and explain it. Okay. Yeah.
Yes, sir.
Hello, can I speak?
Yes,
Okay.
please go ahead.
Okay, I asked the question on the database he used because whatever you are publishing should be reproducible.
Exactly.
That's one thing.
Yes,
That's one thing with molecular biology. I mean, computational biology. So, though I have not read the paper, but that's why I was so keen knowing the database he used, because most of those SDF formats are readily available for ease of reproduction. Thank you.
Yeah, thank you very much, sir. Thank you very much, sir. So it's true. That's the thing. PubChem database is like my go-to. If I'm working on any Nigeria plants and I try to access, if I get the list of compounds, my go-to website is PubChem. So any compounds I can't find, I use ChemDraw, which is another software to draw it and convert it into SDF formats. But for this traditional Chinese medicine, we're given a job to run it, to check the bioavailable compounds. And I tried working on it with TJ5, and that's it. So I'll try to confirm the source again for my professor, sir.
Okay.
Can I ask or make a comment?
Okay, sir.
Okay.
You didn't tell, although I've not read the paper, did you declare
sir.
any conflict of interest?
I did not declare any public of interest, no competing interests. The authors did not have any competing interest, sir.
No, because you said you got it from a professor. Your professor may have gotten it from
the professor.
So
the professor was on the paper. The professor was on the paper, sir.
Okay, thank you.
Yes, sir.
Okay, so as part of your conclusion, was there any recommendations for future work? What were the shortcomings of the work? And then what do you recommend that could be done to mitigate those shortfalls in your work, in your own work? I know we do not have much of molecular laboratory setups in our continent. So based on your in silico work, what would be your recommendations? And anybody want to carry out a similar study, what advice would you give to such?
So in silico works, the major advantage or the real reason about in silico techniques are just like a setup to any in vivo, any molecular work, So now I think basically I feel the highest recommendation, the highest quality of job has been done in this publication because the reviewers were kind of strict and they wanted like the best. So, and I think using the molecular simulation is like the best of the best in any computational review. So, and 100 nanoseconds, which is kind of like the pass mark. So 100 nanoseconds. So based on the results of the 100 nanoseconds molecular simulation, it is a very, very good compound. So I think the next thing and the limitation, which I faced from one of the first journal I've submitted to, which was Chinese medicine. Then there's an Elsevier journal, Chinese medicine. So the first thing they told me was I should do an experimental data. I should try to carry out this maybe in vitro. So I think that was like one shortcoming. So I feel the next thing I should do, or the next thing someone should try to do, is try to synthesize this compound and try to check them in vitro, in vivo, and try to check the efficacy. So if it is working, or if there are some shortcomings, then maybe editing and modifying the compound structure should be the way forward, sir.
Okay.
Yes, sir.
Okay.
I think we can come in here. That is why we are on the research hub, and maybe probably we can collaborate together with anyone that is interested. We can do a laboratory further work on this. And so if you are interested, you can just reach out. Dr. Kojo.
Yes. So I was going to ask, in molecular docking, one of the obvious things we normally look out for is protein folding. Was there any traces or any incidents of protein folding?
No, sir. No, sir. So the protein used is just an X-ray crystallograph. This thing. Okay. It was derived based on X-ray crystallograph. So from PDB. So I just accessed the protein. I got it from PDB and imported it. So no protein folding, no protein modeling, nothing at all. Just the straight structure. The structure we got from the lab, or the structure they crystallized in the lab. This was the structure I worked with. So.
Okay. All right. Thank you. That is all my questions.
Thank you very much, sir.
Congratulations on your work.
Thank you very much, sir. Thank you very much, sir.
Please, anybody with questions, comment, contributions, or
well, I just want to commend Bodun on his passion about research, even at the level of being an undergraduate, and you are doing so well. You are doing a lot. I want to commend you, and I want you to keep it up. Well done. And I see someone chatting me, Dr. Florence. We can, if you are on the research hub or on the journal club, please, can you just reach out to me on WhatsApp? Okay, I can see your, you can just chat. I will send my, maybe my email or my number on the WhatsApp, on the chat. So you can just reach out to me directly. Or if you're on the AutoHead research hub, you can also reach out. So let's see how we can collaborate with other team in the research hub. So thank you.
Okay. All right. So any questions up to this time? Any comments, suggestions? For those of you who are not on our Telegram platform, I've shared a link in the chat box, so click on it and join us. All right, so I would like to hand over to our boss if there are no other questions or comments. Thank you very much, Demi, and your team, and congratulations on your work. Dr. Fumila, if you are here, please take over and then close us. Thank you.
Good evening, everyone. Dr. Chukwujo, I don't have to take over, but this is a very interesting study. I have also learned one or two things, and I want to congratulate Bodun for a good job. I'm quite impressed, actually, by the level of achievement that he has, being an undergraduate. This is commendable. Well done. And we are all open to collaborating with you to doing further studies. So over to you, Dr. Kojo. Thank you for coordinating this session. And thank you, Dr. Aneyo. Thank you, Dr. Ravi. Mr. Felix, you're welcome. And thank you, everybody.
Thank you.
Thank you,
Okay.
thank you, Dr. Fumi.
Thank you, our boss. So I thought Dr. Felix was actually going to give us more exposition and in-depth analysis on the statistics, but I think maybe today, but in most of our discussions, most of our review sections, Ferris lead us in the statistics so that we can improve on text selection as well. So without much ado, I would like to thank you all for participating and also contributing in various ways to make the section what it is. We look forward to welcoming you in the next section. However, between this time and before the next session, there are a series of coming up. Watch this space and look for our Telegram platform, the WhatsApp platform, hosting upcoming events. Do participate and also share within your network and invite your colleagues to join us whilst we learn together. The essence of the review section is to help you and I to hone our reviewing skills. Okay, so we'd like to thank you for participating and wish you a very good evening and a restful weekend ahead. All right, bye.
Thank you,
Dr. Kojo.
Thank you. Bye-bye.
Thank you all.
Thank you for the session.
Thank you, Dr. Kojo. Thank you for me.